Now I would like to invite Dr. Debojyoti Chakraborty from CSIR IGIB India. Deb came up with an innovative diagnostic test for COVID-19 and named it feluda. Feluda is a fictional detective character from India. Let's hear more from Deb about his innovation. Take it away, Deb.
Hello everyone. My name is Debojyoti Chakraborty. I'm a scientist working at CSIR Institute of Genomics and Integrative Biology in New Delhi. And I'm very happy that I have been given an opportunity today to present before you some of the work that is going on in my lab in the field of CRISPR-Cas technology, and particularly in the way that we have been using this technology for SARS-CoV detection. So today I'm going to talk to you a little bit about how we have repurposed the CRISPR diagnostics for detecting SARS-CoV in patient samples. I will start by introducing you to the major detection mechanisms or the major techniques that are being used currently for COVID-19 detection. And as you can see broadly, these can be divided into two major tests. One is based on nucleic acid amplification, where one is trying to detect the viral genetic material directly, so it kind of gives us an impression whether the patient is positive at that current time. And the second one is an antibody test, in which a previous infection to the virus is detected by the presence of antibodies that the body has generated against the viral proteins. There is also a third test, which is called the antigen test, in which the test tries to detect the presence of viral proteins within the serum or the plasma. Now, on the right side, you see that how these tests are generally performed. And in general, the molecular test, which involves nucleic acid detection, relies on identifying the viral nucleic acid, in this case the RNA, making several copies of this RNA after converting it into a DNA, which is called the complementary DNA. And then the gold standard test, which is called the quantitative PCR test, actually uses a very sensitive probe, which is fluorescently labeled, to specifically bind to a sequence in the viral cDNA and then emit a signal. And this is generally the gold standard test. In the case of antibody tests or serological tests, the antibodies in the blood that have been generated due to the exposure to the virus is detected on a lateral flow device, as you can see on the bottom. Now, both these technologies are widely used and they have their own advantages and disadvantages. While the antibody tests do not talk about the immediate infection, it does tell us important information about the population in general and helps in taking policy decisions. But the nucleic acid amplification test or the RT-PCR based approaches seem to be the most suitable to identify patients who are currently affected and then quarantining them. They of course use the quantitative RT-PCR probes and a dedicated instrument which can detect the fluorescence coming out of these QRT PCR reactions, and hence although they are very useful and sensitive, they need the presence of a dedicated instrument which can actually do this kind of experiments. And in a lot of low cost settings or in a lot of developing countries, such kind of instruments are not available. So keeping all this in mind, we thought of actually using a CRISPR based test which would not be needing such kind of a dedicated instrument and can be done at a much more cost-effective manner. So CRISPR is of course precise, simple and robust, and why these properties are a characteristic feature of CRISPR is something that I'm going to tell you in the next few slides. It would not be possible to talk about how we came up with the idea of CRISPR if I don't introduce a little bit about the work that we generally do in the lab because the development of this feluda technology, in which we are actually detecting SARS-CoV, is something that has been a kind of offshoot from work that we have been doing in the lab for the last 2 to 3 years on sickle cell anemia and correcting the mutation for sickle cell anemia using CRISPR-Cas9. So on the left you see a cartoon showing how a Cas9 complex remains bound to DNA, and the blue nucleotide sequence, or the blue part of the cartoon, actually tells about a guide RNA which complexes with Cas9 and then opens up DNA double helix, which is shown as the double helix at the bottom, and then it base pairs, and if it base pairs completely, then there is a double-stranded DNA break, which is then repaired by the cellular machinery to give rise to different outcomes of repair. If the repair happens in a kind of a quick and dirty fashion, then there are deletions and insertions that happen. If it happens in a very precise manner, then a specific DNA sequence can actually be integrated at the target. Now, off-targeting and double-strand DNA breaks, of course, convolute gene editing for therapy. This is pretty well known. And this happens because the 20 base pair of homology that the guide RNA tries to find on target DNA might exist partially at different parts of the genome. And these sites can also be equally bound, and therefore there can be a possible editing happening. And the research all around the lab, all around the world, over the last few years have been to how to develop better editors for therapy. It seems there are two, major ways by which one can do this. One is to actually engineer the existing ones, and that is why by substrate linked evolution or by rational design, etc. Large number of evolved Cas9s or engineered Cas9s have been described. Alternately, one can look at nature and try to see if there are Cas9 proteins which might actually perform better than the existing ones, and these are also Cas9s that have been characterized. So we have been working heavily, like I said, and the work that I'm going to show you is actually done in collaboration with my colleague Dr. Shobik Maiti, and we have been working heavily as a part of the CSIR sickle cell anemia mission to try to find out proof of concept methods to correct the sickle cell anemia mutation in Indian patient derived stem cells. And to cut a long story short, our results were published last year in PNAS, and what we did is that we extensively characterized an orthogonal Cas9 from Francisella novicida and used it to correct the sickle cell mutation in patient derived induced pluripotent stem cells. Now this is an important slide because it tells you that this particular Cas9 had some very interesting properties which we later on went and used for the purpose of CRISPR based detection. If you look at this Cas9 complex carefully, you will see that the protein, the part with which it interacts with double-stranded DNA, also circled as in the dotted circle, is highly electropositive in nature. It has a high positive charge, which actually renders this Cas9 into kind of it basically makes the Cas9 switch off from its binding state to a non-bound state whenever there are mismatches that it encounters in that DNA RNA heteroduplex. And this is a very peculiar phenomena since similar kind of activity is not seen for even high fidelity Cas9s which have been engineered, meaning that this Cas9 from Francisella novicida is highly specific in the way with which it interacts with its substrates. And this was our major finding, what we obtained from that from this study, and we thought that we can actually use this principle of binding to a target or disengaging from a target with high specificity or precision in developing a detection tool. So CRISPR based diagnostics and innovative solutions for genotyping is not something new. It has been in vogue since 2016-17 when the first papers describing Cas12 and Cas13 came in was reported. And a number of companies in the past have been actually started to look into the possibilities of using a CRISPR based detection approach. Most of these CRISPR systems, or the Cas12 or the Cas13 systems, use a transcleavage activity of reporter molecules when the substrate is bound by the Cas12 or the Cas13 proteins. We decided to call our system as FN-Cas9 editor linked uniform detection assay, or feluda, because we already have a Sherlock and a detector system which has been reported, and these are detectives. So the name was inspired by a detective figure from it's a fictional Indian detective made famous by the author and movie director Satyajit Ray. So how does feluda work? So feluda works on the principle that if you have an FN-Cas9 protein, which like I said previously, has a very high specificity to its targets, both in terms of how it binds and also in terms of how it cleaves the substrate after binding. Once this Cas9 protein actually interacts with its target, if it encounters more than one mismatch, then the Cas9 actually evicts from the target. The net result is that there is no binding or cleavage. However, if there is a complete match, or at least one mismatch in the bubble that you show on the left top, then the outcome is that there is binding and cleavage. And we figured out that these outcomes can actually be represented in different formats. One is by a simple in vitro cleavage reaction in agarose gel, which can also be represented as a capillary electrophoresis peaks. Alternately, if one finds a way by which the Cas9 protein can be tagged with the fluorophore, then the presence or absence of the Cas9 protein in the supernatant can give rise to a fluorescent readout. And finally, a lateral flow readout can also be obtained by developing a chemistry for paper-based detection. So we took the example of the sickle cell anemia mutation, and as you can see, the wild type represents a normal blood cell, and the sickle cell, the blood of course gets sickled. And interestingly, this is caused by a point mutation in the beta globin gene, and the presence of the mutation is shown in red, the GAG, which is getting converted into a GTG. So we designed guide RNAs with keeping this mutation at the second position intact. The guide RNA is of course reverse complementary, so therefore there is an A. And then we walked along the whole length of the guide RNA, and we found a position which would be a 2 and the 6 base pair mismatch with the target, where it would produce a completely opposite readout on an agarose gel cleavage reaction, where the wild type substrate would be almost completely cleaved, whereas the sickle cell substrate would be almost not cleaved at all. And this diametrically opposite phenomena of negotiating with the substrate was something that we wished to take forward and use as a readout. So this was a format in which you can actually see it directly on a agarose gel. But we questioned ourselves that this format of cleavage, was it also faithfully being replicated in terms of binding to the substrate? Meaning whether the Cas9 was not bound to the wild type substrate with 2 mismatches and was bound to the sickle cell anemia substrate with a single mismatch. And we observed that this was indeed the case when we did a single molecule binding experiments. This is a bulk molecule study actually using fluorescence MST, or microscale thermophoresis. We found that whereas a point mutation or a single mismatch remains, the Cas9 remains bound to its target with a relatively strong binding energy. The KD is 187 or so. If one introduces a second mutation, then the binding is extremely weak or not existent, and it becomes high micromolar range. So we figured out that this can be utilized in a detection scheme where if you would tag the Cas9 protein with a fluorophore and you would label the targets with biotin by putting biotin-ylated PCR primers, then bound Cas9 proteins would not would mean that the supernatant would be devoid of such fluorophore particles, and therefore would have a lower signal, whereas unbound Cas proteins would be present in the supernatant and would be detected by a detector. And in this way, using fluorescence, one would be able to distinguish whether a substrate has a point mutation or not, as is shown in the right. What we also figured out was that the nature of cleavage or the nature of binding was happening stoichiometrically, meaning that if 50% of the molecules had a point mutation and 50% did not, we would have 50% of these molecules being bound by a Cas protein and getting cleaved, whereas the remaining 50 would not. To understand this, if this was happening robustly, we actually did this in the case of sickle cell anemia patients and carriers who are heterozygous for the mutation. And the pattern is shown on the left here, where you see that the wild types is not cleaved, the heterozygous is cleaved half of, and the homozygous is cleaved nearly completely. And this is not just happening as a one-off event in some cases, but we did a blinded study with at least 50 different samples from a village in Raipur, and the Sanger sequencing was done in a different lab, and feluda detection was done in our lab, and we found 100% concordance between the two results, suggesting that this method of identifying carriers is highly robust. Finally, we figured out that this 2 and 6 mismatch positional identity with respect to a protospacer adjacent motif or a PAM would limit our detection ability in terms of the number of possible Mendelian disorders we can target, because not all SNPs would have a PAM in the vicinity. So by putting a PAM site in the biotinylated primer itself, one can actually trick the Cas9 into believing that there is a PAM within the amplicon, and in this way one can actually target all possible single nucleotide polymorphisms in DNA and RNA that is present in the genome. We showed how this works in gastric biopsy samples containing variants of Helicobacter pylori, and as you can see over here, these variants are different from the normal Helicobacter pylori by just 1 or 2 nucleotides. However, they have a different antibiotic resistant properties to rifampicin, and doctors generally are not able to identify these unless they take a scrape from the stomach, and then they culture the bacteria, and after 2 to 3 days they are able to identify if there is an antibiotic resistance variant or not. Feluda can do all this within 2 hours, so that really reduces the time for diagnosis as well as for effective treatment. Finally, this is not just limited to the lab. We took feluda to a village in a primary health center, as you can see, in Raipur. And in India, these kind of initial tests to identify sickle cell patients or carriers, one does what is known as a solubility test, which is a very crude test, but it gives a kind of an quick and dirty outlook whether there is any kind of mutation present. However, it's a very unpredictable and often inaccurate test. And what we also saw for that this requires the presence or the need to prick blood, or to prick the finger tip and get some blood. And if you look on the right picture, on the right side, there are these students who are very young students, and some of them are looking with not such a very enthusiastic face, thinking of how the blood would be taken in order to do the solubility test. So what we came up with idea was to have a method by which these children could be asked to simply spit into a small tube, and then there would be reaction components present within that tube which would isolate the DNA from the saliva just by leaving on the benchtop for some amount. These are classical molecular biology methods, and then this can be a substrate for the feluda reaction, so making it also possible to do this in a completely non-invasive manner. And as a final step for developing the feluda, we wanted to make something which would be almost be possible to use at home, such as a pregnancy strip, and one can actually use a feluda assay on a similar kind of a strip to detect the outputs. So to do that, we took help of a commercial strip in which there are these zones that are marked in different colors, and the bottommost zone contains an anti-FAM antibody conjugated with gold nanoparticles. So if a sample has got FAM in it, FAM label, then the anti-FAM antibody gold nanoparticles interact with that, and it takes the sample up the strip, and then if that sample also contains a biotin label, then there is a streptavidin band which is shown on the extreme right with a very dark red area. This streptavidin biotin reaction gives rise to a signal there, whereas if the substrate did not contain a streptavidin, then the entire complex moves up and there is a control band region where a control line will be formed, which tells if the strip has worked fine. So we had to bring in the FAM at some point in the CRISPR complex and also make sure that our substrate was going to be labeled with biotin. This we managed to do by doing a PCR in which the primers were biotin labeled. And in order to make the Cas9 RNP FAM labeled, we made a chimeric guide RNA where the tracer RNA sequence was now going to be labeled with FAM, whereas the CRRNA remains intact without a label. And we systematically figured out what would be the optimal length of the CRRNA tracer RNA overlap, because this was a new Cas system, and we found that a 19 nucleotide overlap would retain the properties of Cas9 RNP complex formation on a target. And using that, we could go down to almost femtomolar levels of concentration and was able to detect even a single nucleotide polymorphism by the presence of a band on a strip. So this was where we were at the beginning of the year, and we thought that we had a nice method by which we can detect single nucleotide polymorphisms. But of course then COVID-19 struck and life turned upside down for a lot of us. And we quickly realized that this feluda assay can be also very useful for detecting the COVID-19 pathogenic sequence. And we developed a technology around that, where the samples that are collected and the RNA is extracted from these samples, and on the step 3 can go into a single step RT-PCR reaction. Now the RT here actually stands for reverse transcription reaction, not the real-time PCR. So this means a regular PCR instrument or a regular PCR machine can serve the purpose. And the reverse transcription PCR is done using two primers which are biotin labeled. And this is followed by the lateral flow assay, in which the FAM labeled Cas9 RNP complex is targeting 20 nucleotides of the viral RNA sequence in that amplicon. And if it finds that, then there is a positive band. If it doesn't find that, then there is a negative band. This has 3 layers of specificity because we use first a reverse biotin labeled primer specific to a COVID gene. Then there is a forward primer which is also specific to that same COVID gene. And finally, the high specificity of Cas9 RNP towards that amplicon, 20 bases in that amplicon. The entire process can be done within an hour. It is economical because it doesn't use the fluorescent Tachman probes, which are the costly element in a quantitative PCR. And it also has a semi-quantitative flavor, which I'm coming to it in a minute. And there is no requirement of specialized equipment. Just a simple PCR machine, which is present in schools, in universities, in healthcare settings, can be used. And as you can see, this is a slightly older slide. The starting RNA amount, to put it into rough estimates, one can detect starting up till 10 copies of a target RNA, and CT values almost going up till 40 using a feluda detection. And about an hour it takes if one uses the single step cDNA preparation and a normal PCR. If one uses an RT-RPA or isothermal way of developing amplicons, then this entire process can be brought down to about 30 minutes. And at a lab level, this costs roughly about 600 rupees in the Indian rupees. And the final readout looks a little bit like this. There is a positive control which gets a band and the sample, whereas a negative control and a negative sample will not have any of these bands. And this happens by lateral flow. So if you put these strips in a PCR tube, within a couple of minutes, as you can see on the leftmost on the strip, there is the control band developing first on the top, and then you can see the test band developing at the bottom. So it's a pretty rapid test. And we also developed an AI app which we call as Topshe for people who have read Satyajit Ray novels on feluda. They know that feluda is often assisted by his cousin who is called Topshe and a third friend who is called Jatayu. So our AI app was called Topshe, which is true outcome predicted by strip evaluation, which can be used to take a picture of the strip, and then it does background correction, normalization with the NTC control, and then can give a readout based on the training on machine learning done over several hundreds of samples. What is also important to understand here is that the feluda reaction happens by stoichiometric binding, which means that every molecule of the target is bound by a molecule of FN-Cas9. So if there are 50 molecules, there would technically be 50 molecules of Cas9 binding, and there would be a corresponding strip intensity. So we found that there is a pretty high concordance between the qPCR and CT values and the feluda intensity. A low qPCR CT corresponds to a higher feluda band intensity, giving it a semi-quantitative flavor. So one can detect whether a patient is highly positive for COVID-19 versus a patient who is probably just recovering with a very low weak intensity. This is slightly different from how the Cas12 and the Cas13 based systems work, where there is a transcleavage activity resulting in a lot of fluorescence signal even from lower number of viral copies. Of course, it has the advantage that it amplifies the signal and therefore can make it into a detectable format. Finally, like I said, that feluda is assisted by Jatayu. We also have a Jatayu, which is a web server available at jatayu.ijib.res.in, where one can put in their sequence of interest, and the feluda guides and the feluda primers will be designed using this tool. So where do we go from now? As you can see that this is still a little bit old. It was from pretty early time point. The number of cases in India and across the world is huge at the moment, and we still rely on the fact that there is sample collection and then RT-PCR or other techniques, and then whether one can identify whether a person is positive or negative. So we have come up with innovations where there might be a possibility to do the feluda test at home using the minimum most of requirements. As you can see over here, it doesn't require equipment, simple workflow, and early diagnosis. This test is also ready. For IP reasons, I'm not able to show how we are doing this. But of course, home testing can also have other ramifications, sometimes even going beyond biological but more psychological and so on. So all these things have to be taken into consideration before launching these kind of tests. But technically, it is possible to advance feluda for doing home testing at some point. So with that, I would like to thank all of you for giving me the opportunity to present before you today. And there are a large number of people who, it's a big lab, it's a relatively big lab with a lot of collaborators. And we are very generously funded, including also the Merck Young Scientist Award last year. And of course, Tata is our official partner for taking this kit. We have licensed this technology to Tata Sons. And the most important slide is actually this. We kind of started working on COVID-19 for feluda at the end of January, expecting that this would become a pandemic. And by March, we had a working prototype which was ready, and by May we could license it to Tata Sons. Currently, it is undergoing regulatory evaluation, and we hope to have this done as quickly as possible, hopefully by the end of this year. And all this work would not have been possible if we didn't have highly dedicated and highly motivated and passionate individuals who are students, postdocs, technicians, and research assistants, and so on, working full time over the last 3 to 4 months or even more, and spending all their time and energy during this pandemic period to come up with a technology that might be useful in detecting COVID-19 and helping people. So this work is actually dedicated to all of them. Some of them turned COVID positive on the way. All of them are doing well, of course, but it shows a little bit about the strength that the student community brings in doing good science, and I'm really thankful to all of them for the efforts that they have put. With that, I would like to thank all of you and would be happy to take any questions that you might have.
Thanks, Dave, for sharing exciting information about feluda, and best wishes from Merck for commercialization of your test.